Welcome to Part 2 of Chapter 5, Membrane Transport and Cell Signaling for EFB103. Let's get started. So in this part, we're really getting into now cell signaling. And that might be when you think about a multicellular organism, all the kinds of communication that needs to occur between cells, or even single-celled organisms more interacting with their environment, receiving signals and interacting with their environment. Multicellular organisms also interact with their environment, but then they also have to communicate between other cells. This works for eukaryotes, and that's what we're going to focus on, but I've got this bacterial culture plate. We know now that even prokaryotes, like bacteria, release molecules into their surroundings. Now, if you take an image like this and you see this cheetah chasing this gazelle here, you can ask yourself, well, what kind of cell signaling is happening in either one or both of these organisms? There's a lot of things that are going on, right? So there's a lot muscle contraction. So there's nerve cells interacting with muscle cells, vision, so light collection cells, and that information is being sent to the brain to be processed. Vasodilation, vasoconstriction, hearing, that all involves cells and cell signaling. And then if ultimately that predator is successful, there's a whole bunch of cell signaling and communication that's going on during digestion. So the basics of cell signaling involve some kind of signaling molecule that's called a ligand or ligand, and then some kind of receptor protein. Now that might be on the surface of the cell like we see here, or if that signaling molecule can pass into the cell, those receptors may be inside of the cell. And there's a few kinds of signaling here, and we can categorize them based on their proximity, really. So endocrine signaling, that's going to be long distance. Paracrine, para means next to, so that's sort of local signaling. Autocrine, auto means self, so this is a cell signaling itself. And then contact-dependent signaling, so the cells actually have to be touching for a signal to occur. So endocrine signaling first. This would also be called hormone signaling. So think of this as that example where a hormone is released by one kind of cell. So here's an endocrine cell. It's releasing these hormones into the circulatory system, and those move somewhere else in this person's body, and the target cells are far away. They're distant. That doesn't mean some target cells aren't also close, but that the signal can spread far and wide and interact with all kinds of cells. So that's endocrine signaling. Paracrine, remember para means next to, so here we have a cell that's close. This is also known as local signaling. So here are these signaling molecules released from this one cell, and here's the receiving cell. These signaling molecules bind to a receptor, and then, as we'll see, something happens. And so paracrine signaling involves local regulators, not hormones in this case. And here we see the signal being released, and here's this adjacent target cell. A special case of paracrine signaling, we give it its own category, is synaptic signaling. Now, again, really, if you look at this is just paracrine, right? You've got two cells next to one another. But here we are talking about neurotransmitters. So those are our signaling molecules, little neurotransmitters. One cell is releasing those, and then that signal is being received by this second cell, which is then stimulated. So this is nerve signaling. Autocrine signaling, this is a case where the cell is sending a signal and it's also receiving it itself. Now that might mean neighboring cells are getting this signal too. It doesn't just mean it's only receiving it itself. But if the cells can receive their own signals, that might stimulate certain pathways in that particular cell that's also releasing the signal. And then contact-dependent signaling. Here we see the cells in direct contact. So here's two proteins on the surface. Let's say this dark purple one. Here is the receptor. Here's sort of this signaling protein, if you will. It's not considered a ligand at this point. But it just involves direct contact, and then the signal is transmitted. And so another kind of direct contact is something we've already talked about, but gap junctions and those plasmodesmata in plants, where you actually have continuous cytoplasm. So signaling molecules can pass from one cell to another. So this is a special kind of direct contact. Here's the one we've already seen where you've got two proteins interacting, and that is the direct contact we see there. So let's take an example from your immune system. So here's an example of a macrophage. This is, remember, like phagocytosis. Phagy means eating, so this is like this big eating cell. And here it is gathering up these little bacteria. So these cells are receiving signals from the environment that there are bacteria there at all. When they start taking them up, they'll release pieces and parts of them in a very specific way. I'll get into that in a second. But they'll release other compounds into the environment that attract other cells of your immune system. Like, hey, there's an infection over here. Come get over here. The cells neighboring start to swell, so you get this what's called inflammation. And then they'll attract long distance some of these other cells like T cells, for example. And then a macrophage will actually bind with a T cell here to activate it. So this is really important, and there's that direct contact. So when we say, well, what kind of signaling is this? We're actually seeing all kinds of signaling, long distance, paracrine, probably some autocrine signaling, and direct contact. So this is all happening in a very dynamic way. So that's the reality. We break these into categories, but a lot of times more than one is happening. They're not mutually exclusive. All right, quick quiz. Which is characterized by a cell releasing a signaling molecule followed by cells in the immediate vicinity responding? All right, that should be an easy one. That's that neighboring cell thing, paracrine signaling. So here are the steps in cell signaling. The first step is reception. Then that signal is transduced, so there's relay molecules often. We'll see there's sometimes a case where this doesn't happen exactly this way. But these relay molecules pass along that signal, and then ultimately there's some response, right? Something has to happen. Maybe a protein switched on, or a gene is switched on or off, or a protein is switched off, but something happens as a result of that signal. And so you could think of it like this, where it seems really obvious, right? But there's sort of this idea of like the light switch, it's off, and then it receives a signal and something's turned on. Again, it could be the other way around too. A cell receives a signal and then stops doing something it was doing. So it can kind of cut both ways there. So let's talk about reception, that first step here. So hopefully you're already getting a sense of how cells might receive a signal. Here's a protein on the surface of the cell. That's the receptor. Here's that ligand. We're adding a little detail here. This is a polar signaling molecule. That'll contrast the next slide in a second. So this polar signaling molecule binds to this receptor. It's got to fit exactly. Think of like how enzymes work on substrates. It's very similar here. The receptor is only going to receive certain kinds of signals, and then it affects some kind of activation of this receptor, and then the signal is transduced. So here's where that contrast is with different kind of signaling molecules. So a hydrophobic ligand, or something really small, that can cross the cell membrane like we see here. In this case, the receptor is actually inside the cell, not on the surface. And so it's receiving the signal. And then in this case, the activated complex, it doesn't pass the signal along. It actually moves right here into the nucleus. This is the protein that's going to do the next step to actually have that response. This might actually be in the nucleus. This receptor might be in the nucleus. That signaling molecule may need to get in here before anything happens. But let's talk about those cell surface receptors first. So there's three categories: G protein complexes, receptor kinases, and ion channels. Your book really skims over the receptor kinases in this chapter, but they do bring it up again in other places. So make sure you have a decent sense of what these guys are about. But let's start with the first one here. G proteins. Now you might look at this and think, wow, this looks like there's a lot going on here. Just take a moment to look at it and we'll go through it step by step. Here we see this receptor. So this intramembrane receptor, the ligand binds. And what that does is activate the receptor. And so the receptor itself isn't going to do anything except activate this G protein. And so this G protein is found on the inside of the cell. It's made out of a couple of protein polypeptides. And GTP and GDP are important molecules in terms of the activation of this. So what happens when the receptor activates this G protein? You end up with GTP. Think of like, ATP being the currency of the cell. Here's GTP. It's another kind of energy currency. And so this is now sort of charged up and ready to go and pass the signal. And so this G protein will shuttle over, just one part of it really, but like we see from our textbook, it just moves over and it activates this other compound, this other protein. In this case, it's an enzyme, and that's what affects a cellular response. So it's kind of this two-step thing. And see where the signaling molecule is attaching. It's this one, not this enzyme over here. So the signal has to be passed from one thing to another, and it's through these G proteins, and they can activate all kinds of different proteins. All right, another one, receptor tyrosine kinases. So these guys come as pairs of transmembrane proteins. When they receive a signal, the two proteins in the membrane come together. So that's what we see here. And each of these uses some ATP, and phosphate groups from the ATP, forming ADP, they get attached to those kinases. So as we'll see in a moment, kinases add phosphates to things, usually taking it from ATP. Now this protein is activated, and it will then activate other proteins. So those will be activated, and that signal gets passed along. So this is how this works. Signaling molecule causes the proteins to come together, dimerize. They come together as a pair, and off it goes. Another kind here is an ion channel receptor or signaling. So here what's happening is we have ions, lots of them outside the cell, not so many inside the cell. And so this channel is closed, but when a signal is received, the channel opens up. And then those ions just simply are moving down their concentration gradient. So it's opened the floodgates, so to speak. And then that affects a cellular response. And then, let's say that signaling molecule comes off or something else binds in its place. Then that channel closes and maybe these ions are pumped back out to create that gradient again. All right, let's put a couple things together here. When a neuron responds to a particular neurotransmitter by opening one of these ion channels, the neurotransmitter is serving as what part of the signaling pathway, given your choices here? All right, so you had to think about back to like synaptic signaling, these neurotransmitters, they're ligands, they're those signaling molecules. So let's talk about transduction now, this next part of cell signaling. This often occurs in multiple steps, and these multiple step pathways, as we'll see when we get into other topics, can be really helpful for regulation. So a pathway could be stopped or enhanced at many stages, but it also allows for amplification. So you could have a couple of signals, and if you amplify it at every step, one protein activates two, let's say every time, you're going to have an affect a big response. And you could think of this sort of like the dominoes falling, where you've got these different proteins or molecules, as we'll see, relaying a signal. And that's why we call this transduction, because at each step that signal is transduced into a different form. So that message is kind of passed from one thing to another. And something we see very commonly in transduction is this phosphorylation of proteins and dephosphorylation. And that is accomplished with two different kinds of enzymes. One is called a kinase that adds phosphates to things. We saw that earlier with the receptor tyrosine kinases. They themselves phosphorylate each other. So kinases will add a phosphate, and often when a phosphate's added, that's activating a protein. Phosphates can be removed, and that happens using a phosphatase. That's the enzyme that takes off the phosphate. And that, again, often that means it's inactivating or deactivating that protein. And so let's look a little closer at one of these signaling pathways. This will happen in multi-steps through what's called a phosphorylation cascade or a kinase cascade. So we're adding phosphates. So here, signaling molecule activates this relay molecule. That will then, say, activate this protein, which phosphorylates. So this kinase here takes ATP, adds a phosphate to this guy. Now that's activated. It adds a phosphate to this guy. And so they're passing this signal along through phosphorylation. All right, so let's put some ideas together here. What would happen to one of these signaling pathways if the phosphatases had reduced levels of function? What would happen? Right, so now remember the phosphatases, they're the ones taking off the phosphates and basically turning off the signal. So if they're not working, that signal is going to last longer. And so that's what would happen if phosphatases weren't working. Now, it's not just all about phosphatases and kinases and phosphates being added to proteins. There's also these other compounds called second messengers. And this is a nice figure here. We're seeing our signaling molecule, our ligand, that's called the first messenger. So this is like the initiator of that signal. And really, that's why we call these other compounds, say inside the cell here, the second messenger. You got to remember the first messenger is that initial signal. And so these are non-protein molecules. You can almost think of them a little bit like internal ligands. And they are important for often amplifying a signal or switching on other proteins and acting as kind of an intermediary. So here we see our G protein signaling pathway. This activates this enzyme over here. What is that enzyme doing? In this case, it's making second messengers. So it's not phosphorylating something, but it's actually making this second messenger, like cranking out this thing called cyclic AMP. So this looks like a nucleotide, as we'll see here. Here's cyclic AMP, so adenosine monophosphate. So here pretty much looks like a nucleotide, but here's the phosphate. And actually it's called cyclic because that phosphate connects over here, down here, as well as up here on this nucleotide. So it's this adenyl cyclase that cranks out these cyclic AMPs they use ATP to make those. And then a cyclic AMP activates protein kinase A. Remember, what's a kinase? That's something that phosphorylates things. So that's going to now feed into one of those phosphorylation signaling cascade type pathways. The beauty of this, you kind of get a hint of it in this picture. One activation of this adenyl cyclase, it's just cranking out tons of this cyclic AMP. So that allows for amplification. Every one of those can activate a protein kinase A. Every one of those can activate multiple of these other protein targets. So you can see where that amplification can get huge. All right, so if you have this amplification, think about the effects. One ligand can set off a pretty big signal. So are ligands long-lived? Why or why not? Usually we think of them as being short-lived. Now, obviously, this varies depending on what we're talking about, but usually they're not that long-lived because only a few of these can have a big impact. All right. Again, let's connect some dots here. If an animal cell lost the ability to produce GTP, what might happen to its signaling system? Given these choices here, and you pause and think about these, go through each one and decide whether that's possible. All right, so if you can't make GTP, where the cell's going to suffer there is in that G protein signaling pathway. It needs GTP to activate that G protein. So let's look at that third part, stage 3, the response. So ultimately, these signals tell the cell to do something. So that might be something that occurs in the nucleus or the cytoplasm. In the nucleus, that may be switching on a gene or switching off a gene. It may be in the cytoplasm, a protein that's already there is being activated or switched off. And so all kinds of things can happen. And so let's look again at a couple of examples. Here would be an example of a transcription factor being activated. So here the signal's received, that phosphorylation cascade. Ultimately, it switches on this transcription factor. What's a transcription factor? It's something that will bind to the promoter region of a gene and say, turn this gene on, or potentially, depending on the kind of protein this is, maybe it could be turning a gene off too. Another way that we see this work is in that cytoplasmic example. So proteins or enzymes that are already there, they're being switched on or off. So here's an example here, binding of epinephrine to a G protein receptor. All you need was say to 1 to start, right? That amplification. That activates a G protein. You can activate 100 of those for every signal. That activates 100 adenyl cyclases. Those crank out thousands now of GT or cyclic AMP. Those activate protein kinases, cascade, ultimately switching on this enzyme that takes glycogen, that important carbohydrate storage molecule, and releasing glucose. So you receive epinephrine, that's going to release a whole bunch of glucose. So you got that energy that you need in response to that epinephrine signal. Another example here, we're just putting the sugars away. So insulin here, binding to an RTK, receptor tyrosine kinase receptor, switching on that phosphorylation cascade, ultimately turning on this protein. This is glycogen synthase. That's taking free glucose and building glycogen, so putting it into storage. Now, depending on the signal and what cell it attaches to, and whether there are other signals going on, cells can have all kinds of responses. So here we see signaling molecule received. There's some kind of response 1. Another option here is multiple pathways are activated in response to a signal. Here we see a cell getting 2 different signals. One thing is being activated. Meanwhile, the second signal is saying, no, don't do that, or do more of that. So there's crosstalk between these. Here is a case of a different cell with a different receptor. It can still get the same signal, but this receptor is telling that cell to do something different than, say, the cell in response 1. Now, in addition to those 3 steps, reception, transduction, response, you could also add another step, which is termination. Signaling molecule maybe just goes away, it deteriorates or gets diluted. And so the case of a G protein signaling, just for example here, you start to get less cyclic AMP production because that adenyl cyclase is no longer activated. Now protein kinase A is no longer being activated. And again, down that chain, you start to lose that signal. Ion channel, we've already really talked about this one. The signal goes away, the gate closes, and then that cellular response stops. And so these are ways that signal might go away. All right, so what happens in the case of a steroid receptor? Now you got to think about what a steroid is, what kind of category of molecule this is, and what we've talked about so far. We're just going to bring back some topics that we've already touched on. So what's true, given the options, of a steroid receptor? All right, so you have to remember that a steroid is a lipid, so it's going to be hydrophobic. It is something that can get into the cell. It doesn't need to bind to a cell surface receptor. And so the steroid receptors you might find inside of the cell, so in the nucleus or in the cytoplasm. Again, like we've already seen here, you've seen this figure. Here's another example where we have a steroid passing across the cell membrane, binding to, in this case, a transcription factor that goes and turns on a certain gene. And this is just another way of looking at it. You have the steroid, or in this case, it's a hormone. binding to this receptor. And when they bind, this is the activated complex. So it's a one-to-one thing. You don't have that signal amplification like we saw with those other examples. And here we see these guys going in collectively and switching on a certain gene. All right, so that's cell signaling. We'll see you next time.